Thanks, Alex, for this kind introduction. And welcome, everyone, also on my behalf. I got a green name, so I'm also allowed to speak. Anyway, so when Alex put the program together, he gave me this tentative title, Seismology of the Brain. And I liked it a lot because that's my major project. But I also thought it would be good to make it a little bit broader and look to pulsatility in the cerebrovascular tree in general. And of course, with our 7T MRI, MRI doesn't need an introduction, I think, in ICP, because it's in your mission that you use MRI to understand brain physiology. But okay, to start, your heart beats your brain. And well, philosophers knew this for a long time, I think, but I just mean it literally, as you can see it here. This is it's a little bit fake. It's an anatomical T1 scan that we warped with a measured motion field. And note the disclaimer, we amplified the motion by a factor of 15 to make it visible. Otherwise, it's rather subtle still. But yeah, I can look at this for a long time because you really see a nice spatial temporal pattern over the brain. So it's really a well organized pattern that you see. You might say, yeah, why bother? This is the physiological noise I'm fighting off in my BOLT series all the time. There's still a lot of work going on how to acquire imaging fast enough such that you can filter out your heartbeat or deal with it another way, that physiological noise is below the noise levels and you don't need to bother about it. I would like to take a different angle today and see the heartbeat as a signal to the brain. And over the last decade, I think awareness has grown a lot that this is actually the case. So you not only have heartbeat evoked potential, so if you analyze EEG signals in sync with the heartbeat, you see activity in the brain that seems to be a response to the heartbeat. It's an interesting niche in neuroscience field that I want to cover. Another thing is that these beats drive vascular and tissue remodeling by a process that's known as mechanotransduction. If you know what that is, you all know it. If you do exercise and go to the gym regularly, your muscles grow. And that's a response of your cells to purely mechanical stimulus. And there's no genes or barely at least. And you see a nice example here. I believe this is a, so you see here endothelial cells. So these are the inner cells of blood vessels of, I believe, a carotid artery of a rabbit. And if you expose this to normal pulsatile shear stress or shear or flows, you get nicely aligned endothelial cells. But if you have abnormal pulsatility, in this case, it's almost that. You get more or less a mess. And this could be one of the drivers of a condition that's called cerebral small vessel disease. And if you grow older, your vessels stiffen, and more pulsatile energy ends up in your vessels, and then you go probably to the other extreme. That's not in these images, but it could be one of the drivers of the known blood-brain barrier leakage that you see happening in these patients. Another thing is that it is claimed to have a role in brain waste clearance. The brain is a kind of the Western world of the body. It's only 1 or 2% of your weight. It uses 20% of its energy and also produces a lot of waste. And it needs to clean it up to keep your brain functioning well. And now this month, exactly 10 years ago, the heartbeat was really pushed forward as the main driver of waste clearance by convective fluid flow and driving convective fluid flow through a tissue. I think this is still much debated if this is really how it exactly goes. But what you could imagine is that at least something like stirring and mixing of interstitial fluids and perivascular spaces takes place. Like if you dip your tea bag, then the colors and the flavors go out much more quickly than if you just wait for diffusion. So I think there's lots of reasons to study the pulsations in the cerebrovascular tree. And before I go over MRI methods that can do it, I just want to a very short recap of what we are looking at. So here you have the heart, left ventricle, aortic valves, and then during the systolic phase of the heart. And there's a really powerful burst of blood that's being pushed into the aorta. So within less than 300 milliseconds, you push 70 milliliters in your vascular system. And you also see that the aorta is a very elastic artery, so it bulges to kind of engulf that amount of blood. And then in the diastolic phase, the aortic valve closes and your aorta recoils to maintain a more or less steady flow in the end of the vascular bed. And so your capillaries have a very stable flow. Well, that's remarkable given the beating start, basically. And there's this damping of the pulsatility along the vascular tree all the way up to the brain. And if you look to the brain, there's also an interesting phenomenon going on, which is known as the Monroe-Kelly doctrine or principle. And that's actually focused around the observation that the skull is very stiff and that the brain is completely encapsulated by the skull. So there's actually no room for a volume of blood to come in without displacing something else, because all constituents in the brain are incompressible, and the fluids, the tissue. So if arterial blood comes in, something else must go out. And well, of course, there's venous blood that goes out. So what's the problem? Well, the problem is that venous blood is much less pulsatile. So there's a mismatch between the inflow of blood and outflow. And you have to deal with that somehow to not obstruct your blood flow to the brain. And that's one of the roles of cerebrospinal fluid. So there's the ventricles are filled with thicker or water-like fluid, and it's also surrounding the brain. There's many functions, but one of it is also to buffer this imbalance between inflow and outflow of blood. And then the only way out is actually here through the foramen magnum at the bottom of the brain. You can push CSF towards the spinal canal. So it's good to know these boundary conditions more or less to also be able to kind of pinpoint targets that we want to measure with MRI. So and that's actually my kind of passion and also what I like to show here today. How far can we push MRI to measure these pulsatility along the cerebrovascular tree? Now I don't need to explain, I think, that pulsatility influences the blood flow of the velocity, the blood flow velocity and the blood vessel diameter. It induces brain tissue deformations that drive in turn the CSF flow and maybe also interstitial fluid motions. Now, Alex asked me to make it a little bit educational, so I tried to go very high over all these topics and illustrate how MRI and mainly basic sequences can help you to get information on these levels. So let's start with the velocity. So blood flow velocity, you may know that to measure that you can use phase contrast MRI. And the basic sequence of phase contrast MRI is just a plain gradient echo. So you excite your signal for a certain slice, you do your phase encoding and frequency encoding, and this is a very simple 2D phase contrast sequence. Now to measure velocity, what you do is you add a bipolar gradient. And normally you do this in the slice selection direction, so you measure blood flow that's perpendicular to your slice. And the idea is that, well, if you have a static spin, a static piece of tissue, it gets a certain phase, but you rewind it immediately after that, so it doesn't do anything. But if you move, then you get a different phase, rewinding of your phase, and that you get first lobe. And this phase difference, you can show that it's proportional to your velocity. But you probably also know that phase is influenced by a lot of things, such as susceptibility, RF, off resonance, you name it. So there's always a kind of confounder random offset that you want to get rid of. So what you do is you alternate these bipolar gradients, make two acquisitions, and then if you subtract these two, then all constant phases drop out, and your delta phase is then proportional to your velocity. Now, if you apply that to the heart, then you get something like this. So you see here the ascending aorta, the magnitude image, and the phase, which is now actually scaled to a true velocity map. And if you multiply area and velocity, you get your flow rate in milliliters per second. And you see this here over the cardiac cycle. So I already said in 300 milliseconds, you the entire ventricle, or at least 70% of it. And then afterwards is basically zero flow here. Really quantitatively measure that time resolved over the cardiac cycle. Now, okay, so for big vessels, this is straightforward. Now, if we move on to major feeding arteries to the brain, so this is the internal carotid artery, you can do the same. So you see here the mean velocity in centimeters per second over the cardiac cycle, and here the area diameter in square millimeters. And you see here that still the artery is pulsating, so making room basically for the bolus of blood volume that's passing by. And now it's an interesting study by Rick van Tuyl that he looked at 3 different levels. So here you have the carotid artery outside the skull. Here it's passing through the skull, and then it ends up within the skull, so just below the brain. And he measured, one thing I forgot to mention. So if you want to study pulsatility, we used often the pulsatility index, which is defined as very simple, maximum minus minimum divided by the mean of these traces. So he measured at 3 different levels. So below the skull, more or less in the carotid canal or just above it, and within the intracranial volume, so just below the brain. And these are called the C1, C3, and C7 levels. And he measured the pulsatility index for both the area and the velocity. And you can imagine the area pulsatility says something about the distensibility or the stiffness of the vessel wall. Now, what he got was, I found it a bit surprising, but it's not, if you think about it a bit more, but this is just simple 3D data in a lot of people. And what you see is that the pulsatility index is about 0.8, just below the skull. Then it goes up if your carotid artery passes the carotid canal, and it returns again to the same value if you're inside the skull. And what you see also is that the area pulsatility index does the reverse, more or less. And so you see a decrease of pulsatility of the area when it's kind of limited or hindered by the bone of the carotid canal. And that made me realize that normally you often look to pulsatility indexes of the flow curve. Flow is area times velocity. So if you multiply velocity times area, well, your flow pulsatility is the same because there's no way where the flow can go. But there is a difference in how it is presented to the vessel wall, depending on the direct environment or the direct properties of the local properties of the artery. And also, if you, I didn't show it here, but if you make spaghetti plots of each individual, you see in each individual this pattern. So it's not just the average; it's really on an individual basis that you see this. So it's good to realize that you probably should study velocity and area pulsatility separately. Now, this is back to big vessels, easy. How far can we go? And this is already a little bit older work, but still, I think, nice to show. Here we are above the corpus callosum. And you see here, if you zoom in, kind of star constellation of tiny vessels. And those in the white matter are known to be smaller than 0.5 millimeter, so in the order of 300 microns. And you also see negative velocity, and that makes sense because the arteries run on top of the brain and then penetrate, so the velocity is in the feedward direction, which is what we see here. And then if we average over all vessels that we would find, you get a pulsatile curve still. It's around 1 centimeter per second. It's also underestimated because our voxels are bigger than the vessel size, so yeah, it's not perfect, but at least you still see pulsatility in these small vessels. And as to compare, we measured at 3 levels. So in the middle cerebral artery, the big artery at the base of the brain, perforating arteries in the basal ganglia, which are in the order of 0.5 to 1 millimeter in diameter, and then these tiny ones in the centrum semiovale. And then here you see the normalized velocity curve, so you can see the pulsatility right away. And max min minus is the pulsatility index, but it's about 0.6 for the M1, 0.4 to 0.5 for the basal ganglia, and 0.2 for the centrum semiovale. And now currently with improved acquisitions and analysis, this is even higher. But what you see is that the pulsatility does increase if you go to much smaller arteries, but the velocity goes down by 2 orders of magnitude, from 40 to 4 to 0.6, while your pulsatility only decreases by a factor of 2 to 3. So I think there's still pulsatility even in the true microvascular bed, because it's not so likely, to me at least, that from these tiny vessels, perforating arteries onwards to the capillaries, suddenly all pulsatility would be gone. Anyway, so but we can now look at pulsatility, velocity pulsatility, at least in the perforating arteries. And the question is, of course, if we do this in patients, what do we see and what can we learn from that? And I would like to point forward to the oral presentation of Stanley Pham this afternoon, where he shows some of these results when we do this in patients. Now, what about microvascular diameter pulsatility? So we can measure the velocity pulsatility, but I already said these vessels are way below the voxel size, so it seems to be impossible to measure the diameters and the diameter pulsatility. Maybe we can derive something from brain tissue motion. And asking the question is, of course, answering with yes, we can. And the idea is that, so my definition actually, that's also good to mention, of small vessels is just all the vessels that are inside the brain tissue. So it's an arbitrary cutoff, but pathologists might have a way different definition, but it's good for you to know. All vessels inside brain tissue I regard as the microvasculature. If you have a voxel of brain tissue, then the microvessels inside will swell with heartbeat and also make tissue swell. And that gives rise to what's called volumetric strain. And if you can measure that, then you can derive something of the arteriolar diameter pulsation of the small vessels inside the tissue. Now, how can we do that? For that, we don't do velocity mapping, but motion mapping. And motion mapping, that's the second sequence I wanted to for today. That can be done with displacement encoding with stimulated echoes. It has originally been developed by Electros for cardiac imaging, and it's a little bit of a complicated sequence, so I want to give a little bit of a shortcut explanation that hopefully gives a feel of that you indeed how it measures motion and how it can be so sensitive without going into too much detail. So if you start with spin echo sequence, that's probably known. And 90-degree excitation, 180-degree refocusing, slice selection, gradients, frequency encoding gradients, and phase encoding gradients. So this is a simple 2D spin echo sequence. Then we can do a similar trick as for velocity mapping, that we insert two, what I would call motion probing gradients. And now they have the same direction because the 180 actually reverses your phase, pitching the sign of the gradient. So you will also get a phase that's proportional to your velocity. You can feel that probably. It's exactly the same principle, but now with a spin echo instead of a gradient echo. And if you want to get rid of all confounding phases, you again do that with two polarities, if you will. Okay, but normally these spin echo sequences with motion probing gradients are used with such strong gradients that you're also sensitive to diffusion effects. So even if your voxel is standing still, then the water motion inside the voxel gives random motion, and so random phase contributions. Sum that up to one signal, then actually your magnitude signal starts to decrease because you just lose phase coherence. And that's always described by a b-value. So b-value describes basically the strength of these gradients. And then you will see an exponential decay of your magnetization for your signal and the magnitude of your signal. That depends on the diffusion weighting and the amount of diffusion coefficient and amount of diffusion weighting. So now in one sequence, you have both diffusion effects in the magnitude and coherent motion effects in the phase. And now comes the trick. I call it the transformer trick. If you think of this spin echo, the 180 has just two 90-degree pulses back to back, then it's still in 180, of course. So you still have a spin echo. And the funny thing is that you can actually separate these pulses in time by as long as you want, basically. And you still get a spin echo that has the same behavior. The only thing is you lose a factor of 2 in signal for reasons I won't explain. And the good thing is that during this period in between these pulses, which is called the mixing time, your signal decays with D1. And normally spin echo, your signal is 2. This is half your echo time. This is the other half your echo time, so you get 2 weighting. But this kind of extra time in between the two 90s, or in the split 180, if you will, goes with T1. So that allows you to really separate your echo by a long time from, or your motion decoding, if you will, from the motion encoding by a long time, in the order of 1 second, which is T1 of tissues in the order of a second. And that allows you to wait for a very long time for very subtle motions to accumulate phase. And that's why dance is so powerful in measuring these subtle motions. The first time I encountered this was when I was a first-year PhD student 20 years ago by Timothy Rees, and he showed that by doing this, he could measure the strain tensor or the strain rate, because he just had one time point in the cardiac cycle in the brain tissue. And I remember when I saw that for the first time, I was really amazed, because look at the legend. The observed parenchymal strain rates correspond to displacement of 6 microns per voxel with a standard error of 200 nanometers. And this is insane, because your voxel size is about 3 millimeters, and you still can measure displacement in the order of 1 and the size of a red blood cell, basically, at 6 microns. So you should never get used to how motion sensitive MRI in fact is. Well, that's my message for today. Anyway, and so I wanted to use it, but back then I also thought, yeah, who cares about these subtle brain pulsatilities? Who cares, basically? And it took a long time, I think, for all of us to realize that this is the heartbeat is actually a very interesting thing to focus on. Now, one more trick. You can even replace this angle 90 by a lower angle and then do it multiple times in a row. So do multiple decodings of your motion and then get the signal time resolved over the cardiac cycle. And that is what we did here. I still have, well, let's see all references, unfortunately. So it has been implemented on a 3T by Sullinger from the group of Peter Beusker. And we did it again on the 70. And here you see then right-left encoded motion, anterior-posterior encoded motion, that encoded motion, and again you see the quantitative scale bar. This is really, the color is telling you the displacement of the tissue in microns. And I also placed the magnitude image next to it for your reference in the radiological convention. So right is on your left hand, etc. We did another trick actually, that we varied the pulse angle over the cardiac cycle to keep the signal constant. So here you don't see the T1 decay that you would have by waiting longer and longer in the cardiac cycle to decode your motion, and diffusion effects are kind of counteracted by increasing the flip angle underneath. But that's a detail you might forget. So if you look to these motion patterns, what you see that the right side of the brain turns red. So that means leftward motion, and the left side of the brain turns blue. So that means rightward motion. So you see an inwardly directed motion, and it's also the case for the anterior posterior motion, and it's pointed downward during the cardiac cycle. And that makes sense because the arteries run on top of the skull, and then the blood is flowing inward, and the momentum is kind of imposed on the brain tissue. So the tissue is pushed ahead of the blood flow, basically, towards the main opening in the skull, the prime magnum at the base. Okay, so we can measure that. And this is the basis of this nice animation that I started off with. And now when you have these displacement maps, you can also estimate the volumetric strain. So if you compute the divergence of your displacement field, you get your volumetric strain. And for 1D, this is easy to see. So if things don't stretch while moving, then the displacement is the same for two neighboring points. But if the thing is stretching during motion, then your displacement of two neighboring points is different. So your gradient of your displacement tells you something about the stretch or the compression of your tissue. And you can easily show that if you add up all the three orthogonal stretches, you get a volumetric strain estimate for small deformations. Now, so we can do that, and Daisy did that, and we analyzed it for gray matter, white matter, and carefully removed anything that looked like CSF. And then this is here what you get from the volumetric strain curves. Now we start at peak systole, actually, but we use the pulse oximeter for triggering. So you start in the middle of the heartbeat, and here the next heartbeat is coming, basically. And what you see is that, well, if you measure twice, you get twice the same curve, which is always good to see. This is reproducible. And what you also see is that gray matter has a way higher volumetric strain than white matter. It's about a factor of 2 to 3 higher. And that perfectly matches your blood volume. So this indeed suggests to me that what we see here is the swelling of the microvasculature inside the brain tissue. And this indirectly tells you something about the diameter swelling of the microvessels inside your brain tissue. But there's another interpretation possible, which is always the case with MRI signals. You could also think that, well, we pressurize the vasculature for gray matter and white matter in the same way, but if gray matter is less stiff than white matter, it would also inflate more. So this could also reflect stiffness differences between gray and white matter. So we have an ambiguity here on what it means, but at least it's reproducible. And we still, this is also a limitation of the technique. So you see, for example, here some big vessels in a sulcus, that's here. And if those swell, they push these sulci apart, which also would give rise to a apparent volumetric strain. It's not truly the gray matter swelling, but sulci being pushed apart. We try to get around it, but it's not to be excluded. But regarding the ambiguity that we cannot tell stiffness apart from blood volume changes, I ended up with the idea of applying seismology principles to this data. And what you see here is the United States with, on the surface, some measurements of seismic activity of the Earth, and they can use it to kind of figure out what's underneath. So subducted tectonic plate, for example, with different stiffness and different porosity, like it contains oil or gas or whatever. And this is actually the same thing as we want, but then for brain tissue. And I have the benefit that I have measurements throughout the entire 3D volume. So the math is the same, but my measurements are better. So it's a better conditioned system to solve. And this is something Marius will present to you the last day. So you have to stay. You want to see how this goes. Anyway, I'm a little bit distracted. So we could, from velocity pulsations, diameter pulsations, to diameter pulsations of small vessels. And there we got to the volumetric strain. And now we are at the same time at brain tissue deformation. And those two are very closely linked. So back to brain tissue deformation. or for brain tissue deformation, I should say. We perfected the dense measurements a little bit further, such that we now have what we call strain tensor imaging. So we can map the full strain tensor for each voxel in the brain over the cardiac cycle. And strain tensor, you can consider it as 3 eigenvalues and with eigen vectors. And the vectors point in the direction of the principal strains. And these principal strains are just pure expansions or pure compression, so no shears or whatever. So it's fairly easy to interpret. And like diffusion tensor imaging, you sort them from large to small. So the first principal strain is the largest, which is expansion, so the expansion of the tissue with a heartbeat. And what you see is that you have this centrally directed expansion. So right, left is red, anterior posterior is green, and feet head is blue. So you see at the sides here, you have right left expansion, anterior posterior expansion, and feet head expansion. So this nicely inwardly directed stretch of your brain tissue, which you have seen from the movie as well. And then this is the amount of stretch that goes along with it. And you see that in the center, it's more than at the edges. And if you combine it, you can summarize your first principal strain component in one go. Now, the third one is actually negative, which means that you see compression. And that was initially a little bit surprising to me because I thought these vessels are randomly oriented, so you would have a kind of isotropic swelling of the brain tissue. That's not the case. What you see here is actually the Poisson effect. And you know that from stretching a rubber band, you lengthen it, but it also thins in the orthogonal direction. And you see the same happening in the brain tissue. And the second one is in between is basically 0, so we ignore that one. But if you zoom in on the third principal strain, the compression, I find it intriguing that you see patterns that are symmetrical, but not so homogeneous as the first principal strain. So for example, if you have stretch in the anterior posterior direction here, so sometimes it's accompanied by compression in the right-left direction, and sometimes it's accompanied by compression in the feet head direction. And we don't know what it means yet, but first, guesstimate would be, of course, that microstructure and, for example, fiber orientation plays a role here. But we are looking into this, and this is not apparently the case, so it's probably more complicated than that. But it looks like a very rich data set that we still have to get to know better. And to give a little bit of an overview, you see here an average in the MNI space, so that's kind of atlas, over 9 subjects. We didn't do any smoothing, so just average all on top of each other to give a better view of how this first strain and third principal strain looks like over various slices. Now, again, you see the centrally directed stretch quite consistently, but you also see these rich patterns in the compression maps, like subtle purple things that are pointing kind of inward and downward here. And yeah, again, we don't know yet. This looks like, for example, corticospinal tracts, but we looked at it with diffusion tensor imaging, and it's not really one-to-one coupled to the fiber orientation. So there's more going on that we need to figure out later. Now, I said in the beginning that this brain tissue deformation drives also fluid flows. And the first thing that we did in this area was actually a kind of validation study, because if we claim that we measure brain volumetric strain, and then we should be able to match what we see in terms of volume change of the brain with what we see in CSF outflow in the spinal canal, because that's the only way where we can go out. And so we measured that the outflow of CSF here with a plain phase contrast sequence and the volumetric strain by just integrating the volumetric strain over the entire brain. And you see very nice correlations. These are 9 subjects. Horizontal axis, you see the tissue expansion. And again, we are looking to very subtle effects. It's less than a milliliter volume change on a brain of 1 liter. But it nicely matches the amount of CSF that you see passing by. And Lotte will actually talk about this in a patient study where we use CO2 to invoke blood volume changes and CSF flows, and also look how, for example, edema, so tissue properties affect the relationship. Another thing I wanted to focus on is that if you look to the higher extreme, so this is the peak systolic phase of brain expansion, all data points are below the identity line. So that means that the brain tissue is expanding more than the amount of CSF that you see being pushed towards this spinal canal. And that suggests that something else is being compressed inside the skull. So probably veins are squeezed together, and venous flow is also increased and compensates the lack of CSF outflow, basically. And this is something that we are currently exploring, and Alan will give some initial results tomorrow. The final thing, so I think I will stick on my time, Alex, is brain tissue deformation versus interstitial fluid flows. And this is, of course, a very hot topic, if we can measure this. And I once more show you the dense sequence, and I explained already, it's both diffusion sensitive and motion sensitive. So Jacob Jan Sloots designed it such that we have two B values, one of 300, so all blood volume signals are gone, and one of B is 1,000. So from these we can estimate the apparent diffusion coefficient, and from the accompanying displacement maps we can compute the strain rates. And we did it non-triggered, so you see the light blue are the encodings, and the dark blue are the decoding. So this is an interleaved acquisition. So you encode two slices and then decode the same slices right away. There's 100 milliseconds in between. And then if you do this just for all slices for a long time, you get virtually all slices on all moments in the cardiac cycle. And you can estimate both your diffusion over the cardiac cycle and the strain rates over the cardiac cycle, because this is, or incremental strain, we call it, the strain that occurs during the last 100 milliseconds of your measurement. And then if you look, what you find, you see that this incremental tissue strain, which you could consider a strain rate, nicely correlates with the change in diffusivity. So this is the delta ADC, so difference from the mean. So you see the more you strain your tissue, the higher your diffusion coefficient that you measure gets, vice versa. So yeah, this suggests that this brain tissue strain is indeed stirring up the interstitial fluids, because by design we try to get rid of blood volumes. But I still, the most critical myself, we can still think that, for example, plasma in the microvasculature is still too slow to be completely spoiled with the BS300, and that we still look to subtle microvascular flow phenomena. So this is still to be sorted out, what really is the biology below these observations. But it's at least well, consistent with what you would expect if it would be interstitial fluid flow. So with that, I think I've shown you how MRI can be used to track or trace the heartbeat down the vascular tree in various ways. I hope it also inspired you that heartbeat is a topic and that you shouldn't just filter it out of your data. And I also hope that it could stir up discussions and maybe mutual ideas. And now you know that we can measure this. And with that, I would like to thank the PhDs that do the real work, and the funding agencies, and you for your attention.